What you see in this picture is one of the many Facebook data centers, and this one is located in Forest City, North Carolina. In this place, what they do is they store all of our digital information that we post on Facebook in one of these huge data centers. What we can see though is it takes a lot of space and it also takes a lot of energy to keep these and maintain these data centers. So would there be any way that we could try and maybe decrease the amount of space and energy that we use to store all of our information? But there is now. It's called storing our digital information in DNA. My name is Aishi Murali, and I'm going to show you one of the really cool concepts that I learned about recently. So in order to understand how this kind of works, there's some background that you just need to be familiar with. So this is DNA. DNA is what holds our genetic information, and it's one of the essential molecules of life. And so DNA has 4 different base pairs that make up the molecule. It's adenine, thymine, guanine, and cytosine. And adenine pairs with thymine, and guanine pairs with cytosine. And then we also need to know about binary code. So what binary code is it's what holds our digital information. And so it's represented in a chain of zeros and ones, and they can represent our letters, numbers, and symbols. So our current methods of data storage are stored in units called bits, and one of these bits is one binary digit in the binary code, and 8 of these bits make up 1 byte. So in order to understand this experiment, you also need to know some of the studies that have been done beforehand. So Evan Burney and Nick Goldman at the European Bioinformatics Institute came up with this kind of idea, and Dr. George M. Church at Harvard University extended this a bit, and he created some more of a detailed method of how you could probably go about doing this. So what happens is we start out with our words. Here's a sequence of letters that we might want to code into DNA. So what we do first is we convert this into binary code. So that's shown here. And so each of these letters gets turned into 8 binary digits. And this is encoded into DNA right here. That's shown there. And so every 0 becomes an A or a C, and every 1 becomes a T or a G. So that's also known as base 2. Then this sequence is synthesized into actual DNA using some really scientific processes, and so that's how you would encode the letters into DNA. So in order to read this, what you have to do is you have to sequence the DNA in a lab, and so this will give you back something similar to what this looks like, and then that would be decoded back into binary code, which is pretty easy, and then you would decode that back into the letters that you would get in the first place. So that's kind of how the process works. So what we have here is that the mass is per bit is going to be less than our current methods of data storage, because each nucleotide is around 350 AMUs, and each bit for the current methods of data storage would be around 1 million AMUs. Also, theoretically, DNA can survive for about 1 million years, so this would be a really long period that we could store all this digital information, while our average hard drives only last about 5 to 10 years. Like, we have a bunch of hard drive crashes all the time where we lose all of our information. DNA might not have that problem. Also, if we look at the error of encoding and decoding this information, DNA, we don't really know, because we haven't been able to apply this sort of method into normal day life, so we don't really know what the errors would be like, but our current methods are around 3% error. So after doing all this research, I decided that I wanted to try and put my own twist to this, and I wanted to see what I could do with this kind of concept. So I had a plan that I wanted to try and decrease the amount of space that this method would use by even further. So what I decided was I wanted to try encoding every 00 in the binary code into an A, every 01 into a C nucleotide, every 10 into a T nucleotide, and every 11 into a G nucleotide. So this way you would be decreasing the amount of space and the amount of information density by half, so you'd be encoding more information in less space, even within the DNA method. So my hypothesis was that if I synthesize DNA in this base 4 method that I just explained, then it will have a higher information density than if DNA is synthesized in base 2 methods or any current methods of data storage that we have. Also, I wanted to test the percent error that I was talking about earlier. So what I realized is that when you sequence the DNA, the process itself can sometimes lead in errors when you have more than one of the same nucleotide in a row, because sometimes that software isn't able to discern if there are 3 or 4 of the same nucleotides in a row. So I hypothesized that if DNA is sequenced, then there will be more errors when there are 4 consecutive nucleotides than 2 consecutive nucleotides. So in order to test these questions that I had, what I did is that I had created an Eclipse program using Java. And so what I wanted to do is take 100 characters from Harry Potter and the Sorcerer's Stone. Yeah, I'm a nerd. And I created 2 different Java programs that kind of emulate how the whole project would work. So I'm going to run this. And so when I put the text into the program, this is the text, and this is what the binary sequence would look like, and this is what the DNA sequence would look like. This is using base 2 method, so this is what Harvard did. And so then you would see that this is the amount of A's, C's, T's, and G's that you would get, and this is the number of doubles, triples, and 4 in a rows. So then I can do the same thing with the new method, and so if I ran this program, and I put in the text, you would get, this is the binary, and this is the DNA, and you would see that the number of A's, C's, T's, and G's is less than the other one. And then this is the number of doubles, the number of triples, and number of quadruples, and you see that there's actually none of some of these triples and quadruples. So that's a really cool thing, and that could actually reduce errors in the base 4 method. So then we can go back to this. In order to find the percent error in the DNA method, what I wanted to do is try and see how much percent error you would get in the sequencing method. So I chose a known sequence of DNA, and I sent it to a lab for them to sequence it 5 times, so the same sequence 5 times, and I wanted to compare the trials and see what how many errors I would get. So this is what this kind of looks like. It might be a bit overwhelming at first, but what it is really is that I found 2 different errors. So this is in 2 places where there was supposed to be 2 nucleotides in a row, but the sequence came out as 3, and the same thing happened in here and here with the T and the C. So that's in all of this sequence, we only found 2 errors. So that's pretty cool, and that shows that there's pretty minimal errors that might come out of this sequencing method. So these are the results that I kind of got, and so what I found is that the percent error for the DNA method is going to be around 0.053%, so that's way less than 3%, so that's really cool. And I even took this data and I applied it to the trials that I had with the base 2 and the base 4 methods, and I found that the errors were really low just generally. So that's a really cool thing that I found out. So to conclude, what we learned is that the base 4 method is the most effective method in increasing the information density, and also DNA data storage generally does reduce the number of potential errors in encoding and decoding the data that we need to do. So why aren't we using this all the time? Why haven't we been, using DNA all the time? The issue is that it's pretty expensive to synthesize and sequence this amount of information. However, the costs of DNA synthesis and sequencing are decreasing by about 16 times every year, while our current methods are decreasing at a slightly lower rate. So eventually, maybe it will be affordable to use DNA to try and encode and decode our information. So that'll be really cool if we can do that one day in the future. So here are just some real-world examples of how we could use this. So here is the data center that we were talking about before, and so DNA storage could condense this entire server room into a single hard drive that could fit in your pocket. So in the future, we might have issues of trying to find our storage rooms. So that'd be really cool. And also, 150,000 DVDs could fit into a hard drive that's probably the size of your pinky, and also everything that human beings have ever written since the beginning of time, which is around 50 million megabytes. So that could be in a hard drive that could weigh less than a granola bar. So that is some really cool applications of this type of technology that we could have in the future. So yeah, thank you so much for listening to me talk about this. This is something that I really find interesting, and maybe in the future we won't even have hard drives anymore.